Welcome again. Could you please go to the, because we have a bit of a delay, so really good if we can continue it.
Because I like, if I have this, I looks good, doesn't it? It looks good, doesn't it?
Hello. Hi everyone, thank you very much for coming back after the coffee break. The next panel is basically going to address issues such as ethics, ethical ethics, and also the history of art, and the interdisciplinary approaches also involving data science. So we have an art historian, based in the US, and we have, sorry, and like an academic in ethics, and especially in medical ethics. So in order to make the panel, longer, to have more discussion, we're just going to briefly go through. Yeah, you're going to introduce yourself, you're going to talk about it, and then we're going to jump into the discussion. Thanks.
Thank you very much. Thank you for inviting me. It's a pleasure to be here. My name is Joachim Bolt. I'm Deputy Director at the Department of Medical Ethics and the History of Medicine, a philosopher by training. Philosophers don't know how to use technology? So, is that okay? No. Doesn't? Okay, well. So I will talk a little bit about bioethics and ethics of synthetic biology, but not so much, because I also want to say something with regard to aesthetics and synthetic biology, because I actually think that it is remarkable how many artists are working in the field of synthetic biology or collaborating with synthetic biologists, and it is not really clear for me why they do that. So let me start a little bit with ethics and ethics of synthetic biology. As an ethicist, of course, the first thing one recognizes when one looks at synthetic biology that this is a very promising new technoscience, and hopefully we'll see a lot of very useful applications. That's the one side. But then on the other hand, as an ethicist, you are trained in also having a look at drawbacks and risks and dangers, and so that is what we do. We're looking at the possible dangers. So, and the first thing that comes to your eyes when you're looking at synthetic biology is there are a lot of very young, aspiring people, young students, biology students, engineering students, IT students, who call themselves biohackers and who meet in dark places, and they call these places community labs, and they are doing genetic engineering, and nobody really knows what's going on and who these people are. So of course, there are security issues. This is one area that bioethicists like to look at when they're looking at synthetic biology. Security, can there be misuse of this new technology? Where could it be? What can we do in order not to have this misuse? What can we do in order to protect ourselves from misuse? Then, second thing, when you're looking at synthetic biology, you see large companies, Western companies, for-profit companies who are exploiting the new technology, who are developing products, and who are selling these products. And not all of these companies are looking at the results or looking at the consequences of what they're doing with regard to the developing world, farmers in the developing world. These are issues that, for example, the ETC group likes to look at and to point at. So these are issues of socio-economic global justice. And the third point, Of course, as with any other technology, there can be safety issues. Things can go wrong even if anyone who's doing or using this technology is having the best interests and the best intentions. So we could have, with regard to synthetic biology, we could develop, we could produce new organisms, new in the sense that we can put together whole new genomes. It's not, synthetic biology is not only about changing one or altering one genome, it's about altering large strands of DNA up to whole genomes. So what we could end up with is potentially new organisms. What do these organisms actually do? How do they evolve? How do they behave in uncontained uses? These are all questions that we have to answer in order to develop this technology in a safe manner. So that is all bioethics. Does anything of this have anything to do with arts? I don't think so. I don't think so. So the answer is no. So from the bioethicist's point of view, where is the connection between synthetic biology and art? Where do all the artists come from in synthetic biology? And I think one possible answer is the fact that in a sense synthetic biology is, and it has been said many times before, a creative activity. And if you look back at the fact that we can alter whole new genomes, then this creativity becomes perhaps even stronger than or when compared to other technologies, because the perspective can shift from altering parts of nature with regard to certain specific aims towards imagining a nature that is completely different from the nature that we have, nature 2.0. A DNA chemistry that is based on alternative chemical building blocks. All these things become possible, and the one famous quote with regard to these perspectives is, life, the DNA, is 3.4 billion years old. It's time for a rewrite. Also, this, of course, is an expression that conveys the idea that with the means of synthetic biology, we can imagine something new, really basically different from what we know nature is like. And this, I think, that's a valid point to say, well, synthetic biology in this sense is a technology that allows us in the realm of biotechnology to be more creative than we've ever been before. And that doesn't need to be a bad thing, of course. Some good things can come out of it, but I'd like to stress that this kind of perspective is not restricted to specific applications. We might not even know what kind of application can come out of this kind of new organisms, new natures, and still it can be a valid also scientific goal to develop these organisms. Why? Because we can test hypotheses, because we can see whether what we are building actually works. So this would be the interest of a scientist. Is that an aim? Is that an activity that artists can share in? In a way, of course, artists also do something creative. They are creative people, and it has been stressed many times before. But why do they want to be creative? And what are they actually aiming at? And I think the answer must be, well, they have different aims from those that scientists have. Scientists want to test hypotheses, and perhaps they want to build something useful. Artists, what do they want? Well, they want to somehow build something that astonishes us, perhaps, that disturbs us, perhaps, that makes us reflect about the world, about ourselves and our relation to the world, that makes us get into contact with some emotions that we didn't know that we had, or something like that. That is different from the creativity that how science and scientists make use of it. So the question for me would be still, what then is the connection between and the relation between science, synthetic biology, and artists, art on the other hand? And I think, of course, well, it's reasonable to say that both sides can profit from working together in different ways. Synthetic biologists can fill their showcases perhaps with some artistic objects that are nice to look at for the public, and the public then gets less afraid for what synthetic biologists do. And artists can learn new methods, how to come up with new objects, and these objects can be, well, can be living objects. So very interesting for both sides. But does that mean that they are engaging in the same kind of activity? Does that mean that they have the same aims? Does it mean that they are embarking on the same kind of project? And I think the answer to this question has to be no. They're not embarking on the same kind of project. And with any kind of interdisciplinary and transdisciplinary exchange, I think it's important to keep that in mind. Artists are doing something different from what scientists do, and in any sort of collaboration they should be aware of this difference as well. Thank you very much.
Can you hear me? Okay. So I'm going to think we're going to have some interesting discussion because I disagree. But it's okay. My PhD is in art history. That's a quote by Dr. Zoidberg from Futurama, and it's also true. And I'm also a multidisciplinary scientist. And if you look up the Wikipedia page for inter, cross, trans, and multidisciplinary, you figure out that multidisciplinary is the hardest thing because you have to have your shit together in 2 areas. And so that's what I strive for. My PhD is in art history. I'm not a physicist. But that's what I aim for. And so what my goal is as a practice is to do a systematic science of art and culture. And that's where the bioculture, which is in the title of the session, comes in. This utterly looks like a carbon copy of systems biology. And there's more to that than just the iconography. And I want to prove that with an aesthetic product, a figure, which is the lingua franca of everybody in the room. Artists do visual and sonic stuff, products, which don't translate easily into differential equations, which is what physicists do, which don't translate easily into algorithms and the efficiency of algorithms, which is what computer scientists do. And they don't translate into the texts humanists write. But we can all talk with each other by having figures, which, let's be very frank, actually biologists and physicists were not good at either 40 years ago. If you look at PRE papers, like in physics, they have like a black and white line drawing, which usually is very lousy, but now we can actually talk to each other. That's where aesthetics and design comes in. So let me give you one example, and the figure silently also makes a case for that everybody should actually read 6 Easy Pieces and 6 Not So Easy Pieces by Richard Feynman and 7 Easy Pieces by Marina Abramovic, which is sort of as foundational for art as Richard Feynman is for physics. So there is something in performance which is very, very interesting, and what the performance I'm talking about is a performance that goes on for centuries, okay? So as an art historian, classically, what I'm doing is I do a data collection, okay? And that data collection is then subject to qualitative inquiry. And that qualitative inquiry leads to a publication, right? So how can I collect data? I have some data model in mind. And so let's call it DM, so my data model. I have that data model in mind. And I can only have the data model in mind because I haven't collected my data yet, right? So it's Humboldt before he starts traveling. What I have to do is I have to come up with a data model, and what I'm using is formal logic, philosophy. I come up with some stuff, and then I fill it up with things, and the things don't fit. That's the problem. But the key problem between these publications and this formal logic, there's a very long way. There's some dude sitting in a room discussing for 35 years, come up with an ISO standard for data modeling and whatever, it may be Flybase or whatever. It can be humanities or science. And then you come up with all this stuff, and then you follow, in essence, what this is the Ciceronian pipeline, how to come up with a good talk. It's Cicero's rhetoric. This pipeline is 2,000 years old, And what it doesn't allow for is this, is quantitative inquiry. Actually, there's an inhibitor going on. It's very real. Actually, a master's student with me, she learned to program and she does awesome figures, and her advisor said, no, she can't do that. That's not analysis. That's just creative. Major big gorilla in the room, right? So actually, what turns out, we can actually do qualitative inquiry and find interesting stuff to qualify, do quantitative inquiry and find interesting stuff to qualify. Usually this is happening in information science departments because in the humanities department you get inhibited, you can't do it. But what then happens is really amazing. Because we do qualitative research, we can do mathematical models, which I hope biologists in the room now get excited. Okay, you see what this verse is going, actually, because data modeling leads to more data modeling, data collection leads to more data collection, and inquiry leads to more inquiry, and mathematical modeling leads to more mathematical modeling. And I'm just the enzyme who wants to actually get that enzyme going, and it's growing exponentially. The field is growing exponentially, and these guys cannot do anything to stop it. It will happen. Actually, my key point about this is it's already happening. It's not happening in universities, but it's happening at Google, it's happening at Palantir, it's happening at Amazon, it's happening at Facebook. And as a society, we have to be very careful. We want to be ahead and on top of this, because otherwise it will be used to profit, not the society, but only very few people. And so you may ask, okay, what has that to do with art? Cultural production goes on since our species developed a brain that is capable of doing symbolic attribution of stuff on other stuff or meaning to other stuff. And the stuff we produce is growing with the human population. Human population grows faster than exponential. The stuff we produce also grows faster than exponential. And of course, and this is where I would disagree, if we do anything, if Darwin goes out and picks up a piece of algae, puts it in a book, it's a human artifact. At that moment, it is not sort of something that's just happening in nature. Darwin attributes something to it. Actually, Darwin collected that algae and declared it a coral and came up with a coral model of evolution. And then, unfortunately, this other guy came back from New Guinea and says it's a tree. And then Darwin said, okay, it's a tree. But it was still a coral that can merge. So he was more right than he actually admitted to the wider public about the whole thing, because he made a mistake about this algae. And I think that kind of thing, so the history of all human-made things, which art history has been called, this is what I'm after, and I think it should be this way and not the old Ciceronian style. So that's like my premise of what I'm doing and what I'm interested in doing. And I think we can talk, there is some interface. So now the discussion, we talked about bioethics, we talk about putting biologic into culture, and now we talk about the slash, because I think there's a lot of ethics question going on my side too, but there's also on the other side, a question is like, what happens to art if we're living in an exponentially growing world? Thank you very much.
I would like to ask the public if you would like to do a question. Tarsh, hi. Can we get Tarsh a microphone? Yeah, we know.
Oh, wow, yeah.
Thank you. I study in Australia, hence the accent. That's where I'm from. And I'm based at Symbiotica, which is an artistic research group in an anatomy school in a university. And so we work with scientists, we work with anatomists. I'm a microbiologist and a biotechnologist by training, and a artist, and a queer and feminist theorist and philosopher. So is there anything I missed? Physics. I'm not a physicist, although I do use some philosophy of physics. My question, maybe comment, is that most artists know what we're doing. We know it's different from science. Scientists know that art is different from art. I just would like to challenge you on your the value judgments that you were making during your talk. This idea that hackers are, how did you phrase it? Misusing this technology, whereas companies are not. Maybe I misunderstood your implication. I agree absolutely that there are safety issues. For me, as an artist and as a scientist, I think that the reason that I am interested in this as an artist is firstly because there are aesthetics involved, of course, but mostly because there are ethics involved. And I actually want to be able to use whatever tools I have at hand to look at the ethics of those engagements and not necessarily think about whether they're appropriate or not, or whether the methods that I'm using are appropriate or not, or should not be being used. And I think that your phrasing of science as useful and therefore not subject to the same ethical engagement is the implication that I understood from you, which may have been a misunderstanding, and therefore art is distinct from that, is a little confusing. And I'm just wondering if you could engage with the bioethics of synthetic biology as opposed to whether art should be involved in it or not.
Yes, okay, sure. Maybe the presentation was a little bit too short anyway. Because I think, well, of course, if you're looking from an ethical perspective at synthetic biology, what I tried to do was to give some examples that give an idea why safety issues, why security issues, why justice issues play a role when you try to evaluate synthetic biology applications from an ethical point of view. And the biohacker scene in itself, I don't think is very, well, nice people doing usually useful things. So, but the FBI is looking at the biohacker scene also with regard to, yes, of course there is, but the problem is, and that also with regard to synthetic biology as a technology itself, we have a technology that, unlike other technologies, needs equipment that you can buy on eBay or in other places. Small groups can do the work that you want to do. You don't need much training to be able to do these things. So and all these things add up to a scenario, and whether that's realistic or not, it's not, I cannot judge that, but it adds up to a scenario in which organizations like the FBI in the US say, oh, we somehow need to take a closer look at what's going on there, but it's difficult because this is not a kind of technology that can be controlled via satellites, because they, you know, small rooms, small equipment, small groups. And biohackers are example of small groups, small equipment. And from the point of view of the FBI, what would be alarming is who is infiltrating this scene, perhaps? Who is coming? Yeah.
Thanks a lot.
Yeah.
I mean, the same question could be raised also for students gaining PhD or postdoc degrees.
Yeah, but the difference, at least with regard to the US, would be that community labs can be used without, you know, you don't know who's going there. They don't have any admissions. So that would be one difference, perhaps.
I would like to ask a question on those grounds. You've been collaborating with the medical ethics also at the hospital. Please correct me if I'm wrong. Have you so far seen the implementations on regulations, or like until which extent have you seen professionals from the medical field getting worried and eventually talking to the European Commission or talking to bodies that may regulate or may look for a way to control what may be seen as threats, or what may be like, oh look, the Americans are looking at it, maybe we should take a closer look. Have you witnessed so far something that has gone into regulation?
The synthetic biology has not reached, at least not with regard to the physicians that I know in Freiburg, the state, where physicians become aware that there is something new coming, and where they start thinking about what does it mean and do we need new regulations. I think basically the field of medical innovation and clinical research is regulated very well, and I would expect that with regard to synthetic biology, medical applications, we won't need much more regulation than the regulation that we already have.
Max, on those grounds, the spaces where biohackers, we know a couple of biohacking spaces here in Berlin, and we know they collaborate with the biohacking spaces. And what will be also like in the case of iGEM, we know people working in an organized cooperative way for synthetic biology in order to develop solutions. Those places many times, and I've seen myself as I was a member of iGEM for a couple of years. They are approached by artists that eventually go there and they get training, and they want to experiment not only from the technology but also from the perspective of the scientists. No, they want to experiment what is the scientific method, how does it work, and they do come with the illusion of yeah, there's a design, there will be design tools, we'll be able to assemble something that sometimes ends up falling in a functional path, right? We're gonna develop this enzyme or whatever. But as from the art historian perspective, will you see something coming for the arts in that field? If yes, what will be a close analogy in history, some industrial revolution, or what other landmark, what other important time in history could probably relate to these spaces of experimentation?
Yeah, I think there's this twofold answer to this. So on the one hand, and I think that's sort of a common denominator that goes through the entire evening. We have to stop talking artists versus scientists, art versus science. It just doesn't exist. There is moments when we go home and we do some music and we figure out, oh my God, that exponential envelope on that synthesizer, it was like, gives me an idea. I should actually totally measure that at the lab, right? So that's something which is important. That's our roots. Our roots are common. They're in the Wunderkammer. They are in classification. It's not just the same procedure. It's the same people in the same rooms, sitting together, doing the same stuff, only they do shells in the afternoon and they do artworks in the morning or something like that. And I think that is a very important point. And that is also something which can be seen in many, many, many ways in artists and scientists that are alive right now. Let me give you 3 examples, and I want to make that example particular. I thought about this. I'm sorry that I'm another white dude sitting in front here. The audience, for those on the web, is 50-50 male-female. Here's 3 artists, / maybe not, maybe scientists. Fernanda Viegas, she will talk at this other symposium at the Sackler that Roger pointed to. She is an embedded visualization person in the machine learning group at Google. The thing with the bird songs probably was her or one of her collaborators. She thinks figures are the lingua franca. She will actually talk about this. Mariah Meyer, who is a designer at the University of Utah, will actually be one of the keynote speakers at NetSci conference this year in Paris. She was the embedded designer in the Broad Institute. Just look her up. There's a lot of papers that wouldn't have come out without her presence. And then there's people who take it to the uncanny valley of, oh, I don't know what you're doing is science or art, and they get flack. They can only do that because they already have tenure, like Ruth West at University of North Texas, where, she does stuff in data science, biology, and then she goes home and does an art project where they do calligraphy, like Japanese calligraphy, to draw folded proteins. And guess what? You see stuff you can't see in the regular kind of depiction of folded proteins. Obviously, she does this huge art project, she goes home and she does a paper. So that's a kind of way where you cannot really decide who's a scientist, who's an artist. And there's a very long tradition in this, and I think it's very important. Let me tie it back to these kind of biofab labs, biohacker labs. They're like maker spaces for bio. They're all over the US, right? That's the only place where you can do certain types of work. But that does not mean that we're reverting to be homo faber. We're still homo ludens. We're the playing humans, right? Like Huizinga said. And that's true for artists and that's true for scientists. We play around, like most of the time it doesn't work, right? I mean, sorry, I have to admit for me, most of the time it doesn't work. Sometimes it works. But that's what I enjoy doing. And sometimes I have insight, or sometimes I do a drawing that looks awesome. That's cool. So it's the same thing. I think.
Now
Yeah,
I want
please.
to thank you to point this out, because this is a point I also wanted to add actually to what you said, that we should not do this differentiation. And when you ask why the artists are interested in biology, I mean, in the beginning, in the first talk, it is, I mean, the scientist and the artist question exactly the same thing at the beginning, the original point. We have different methods and the outcome looks maybe very different at the end. But I mean, we question about the human being in nature, in the relation to the world, it's political question, it's ethic question, and scientists and artists do the same. They are curious. They want to know about the world, and they step out of the box to look on the things and to find answers in different ways. So it's obvious that they understand each other if they go back to this origin. And you said the playing aspect as well. We like to play, we like to think different, we like to look on things in another manner. And I think it's very important to skip, because we got so specialized in our tools and in our techniques and what you showed. We all have each one will have another drawing, how it has to proceed. But actually, if we drop this, it's very beautiful to think much more can happen. And you just brought 3 examples of 4. But in history, they are all there.
Yeah.
Yeah.
Can I answer one short sentence? I think one important point that we cannot forget is there's now the prejudice in the room. Oh yeah, there's the exact sciences, and then there is the not so exact art. There's a lot of artists like Amish Kapoor, Marina Abramovic, and whatever, who are super precise and exact in what they're doing. It's like James Brown or Prince or Michael Jackson. They're super precise. And as we all know, in biology, in systems biology, have you ever discussed about bias in your data and how stuff doesn't really work out, and there's all these qualitative problems, and the qualitative virologist bullshitting about your high throughput picture? That's exactly the same problem artists have, right? So I think we can't really make these distinctions. Yes, art is a little different than science, but you know, we have the same problems in different ways, sort of.
Ingeborg.
Yeah.
May I comment,
Sorry,
or
go
is
ahead.
that?
Go ahead, Joachim.
Because I mean, yes, it sounds beautiful, and yes, you know, individuals can have different talents, and different individuals can inspire each other by, you know, the one using some artistic methodology and the other one a scientific one. Okay, but still, as a scientist working in the area of a technoscience, and that is what synthetic biology is. At least when you're coming closer to applications, what you have is a certain problem, a certain challenge that you want to use and that you want to solve. And you want to solve it by using technological means, which means that you are looking for an efficient, effective, and efficient solution of how to bring about a certain state of affairs that you think of as useful, hopefully. And that is not what an artist does. I just don't see where maybe that is about, you know, where the origin lies. That is the way you framed it. The origin is somehow identical, and then perhaps it goes in towards different branches or something like that. Maybe that's a way of thinking about it. But I still, you know, when you're looking at an application of a technology, and then you start thinking about certain problems and how to solve them in an efficient and effective way, and that is not what an artist does, as far as I can see.
I don't talk to the people at Disney and
Yes,
Pixar.
Ingeborg.
Yes,
Okay.
I would like to comment on this bashing of the DIY community, because yes, it's true, the FBI had a look into what they're doing, but
Right.
their spaces were never dark.
Yeah.
And this
May I-
is
may I
years,
say
no,
something,
sorry,
because
this
this
is
is
years,
the
and it
second
was just
time-
wrong what you said. This is years and years ago, and we just recently, 3 weeks ago, at MIT Media Lab, we had a huge gathering where Chris Church and others invited the international biohacking community, and by no means this is the I would say the community where the danger is emerging. This is in science. There was a video just 3 weeks ago where Emmanuel Charpentier called CRISPR-Cas a nuclear bomb, and maybe you could comment on that.
Yeah. what I would like to comment on first of all is that this is really a misunderstanding, that it is my perspective on the biohacker community that these are dangerous people and we should be looking at them very closely. I know the German biohacker scene, and they invite me, and I like to give talks to them, and they are very nice and kind people. And I don't have any problems with biohackers.
Yeah, what is your purpose? What is your purpose to comment? What is the purpose to look at that or to comment on that, to stigmatize it, while the danger is in the big corporation or in science? I don't understand that, sorry.
Okay, because I was trying to give an image of the way that, for example, the FBA or political institutions, organizations are looking at what is going on, and the reasons why some of those institutions are beginning to get a little bit nervous. That was what I was trying to do, and maybe that didn't work out. But that was my intention, and I don't personally think that biohackers themselves and personally are any special security problem. I think we agree on that.
I would, yes, just a very brief comment. If we just to build up on your first statement, when we go back home and maybe
can I answer to this?
Yeah, oh yeah, go ahead. Then.
So I think one of the key problems is that the discussion cannot be superficial, right? If we have to understand what's going on, and so if George Church wants to bring back the passenger pigeon and hairy elephants, he's amazing. The groups are amazing who do this. What the broader public doesn't understand, the CRISPR-Cas9 not yet is something you can actually do in a garage. There is some substantial stuff going on, not only to produce the sequences, the delivery is really complicated and hard. So yes, the ethical questions, as you say, arise in the large labs, right? So what gene drives in mosquitoes, for example, right? Have you ever heard about this? So a gene drive is when you basically edit it and then it spreads out through the generations, and then all of the population has that trait. So you could do things that make, for example, diseases worse or stuff like that. So that there is an ethical question that needs to be discussed. But at the same time, I think, yes, there probably needs to be safety standards for hacker spaces, just like there are safety standards for drones. You don't want to have guys on the visitors platform of an airport flying around with drones. It's just not okay, right? And so I think that's, I'm not bashing I don't know, I didn't understand him as bashing.
No, really,
and I think
yes,
the key point, but the key point is we need to think about it, just like we don't have large knives around in a kindergarten, right, to play with.
Anybody? A question? Well, because from my side, I will. If we go back home, or we go back to the biohacking space, and we all basically same. We're not dividing artists or scientists. Please, I'm not saying we have to divide them, but and then we got, say, for example, the extreme case of the FBI looking into that. Then the FBI will be also looking into artistic practice. So is that going to happen eventually? I don't know.
Oh, the FBI looking into artistic practice,
yes,
in
hacking, in biohacking labs. Will that be the case then?
I guess it depends on what artists
are
doing in the lab. I haven't
questions,
heard about that.
Please. Oh, no more questions.
I mean, this is very easy. You should look at the history of the MoMA, the CIA, and re-education in Europe after the Second World War. You don't need to be a bio-artist to be watched. Let's put it like that.
All right. I would like. Please do, yeah.
Hello.
Hi.
I find a little bit dodgy. I understand, and it's interesting to hear these artists, scientists, sort of mirroring that we're doing here, but I think that it's important to point out that the artistic effort differs from the scientific effort, in the sense that the artist is there to basically point out the importance of disentangling, or the importance of separating knowledge from power. I think this is what it's important to be addressed, and this is the whole effort behind the biohacking community. and the question of autonomy, which is basically saying why does knowledge and accreditation associated to knowledge only belonging to one specific community, which is a scientific community. Can we break away from that? Can we make certain practices our own? Can we be autonomous finally from science? So I would say that this is one very important line from an artistic perspective in terms of thinking, right? So it's also the liberty of the artist of questioning this link that it's so present between power and knowledge in the scientific and technological realms.
Yes, I think you're absolutely right, but maybe you have another perspective on that.
Yeah.
So that's why I find a bit dodgy discrediting.
Don't
You know, it's a bit dodgy, like standing on the top of the triangle and looking down and saying they don't have power finally, because yes, we are acquiring power in that sense through the notion of we can be autonomous.
I don't understand you know, there's this thing, you're now discrediting scientists. In your definition, Giordano Bruno was a great scientist because he basically, in recognizing that the universe may be infinite, Earth cannot be in the center, and therefore a Catholic Church cannot be in the center, and stuff like that. So he did. That's a scientific insight, but it has exactly the same effect as, say, you know, some post-structuralist artist who debases some notion of some scientist that the world is like this and that, and you should shut up, right? So I think we just cannot put scientists and artists in two buckets and say one are like this and the other ones are like that. Because you can totally do science the way you describe artists, and you can totally do art the way you describe scientists. I mean, there has been Arno Breker in the Third Reich, Hitler's sculptor. He was not questioning. He was actually doing exactly the thing you just described science to do, meaning perpetuating power by, you know, absolute whatever. And only it was a different system of absolute power. But you know, there is certain things that can be questioned, right? So we have to, in order to talk about the world, we have to agree there is one world. That's what Max Planck said, and I think it's still correct. We have different opinions about it.
Yeah, but just to finish, maybe that's following, because I think the problem here is the notion of knowledge. Because we are in a Western perspective here, and we are accrediting that by following certain standards or certain procedures to be able to create something that is finally recognized as knowledge is a methodology that is sort of imposed and is credited as a certain, by following that, you are credited with a specific power, which is the recognition of a knowledge production, and that leaves away a lot of other fields of also knowledge production.
So okay. So let me make this very straight. I think your argument that what I'm saying is Western is interesting. It's disproved by the fact that the majority of all scientists who have ever lived, because we are growing exponentially as a human population, the majority of all people are not Western. There is a large number of Chinese, Indian, and whatever scientists from all over the world doing science who accept the scientific paradigm, and we cannot just believe it's not true. There is no such thing, because otherwise we end up in civil war. And I'm not saying this lightly. You cannot just say, oh no, climate science is not true, let's just burn all the climate data. We can't do that.
I'm not saying that it's not true, I'm just saying that we can acknowledge otherwise.
But there is no other thing. Global warming is true, you see?
No, I'm not saying that global warming is not true. I'm talking about the methodologies for knowledge production.
I'm really sorry, but we are really running late.
But maybe one way of reframing the issue would be not, you know, that to acknowledge that when we are talking about synthetic biology, we are talking about values also. What is important for us? And of course, there can be very different perspectives. And scientists can have very different perspectives on that, and artists as well. And perhaps some of those perspectives are, in a way, also, well, more typical for one or the other sort of cultural background that one comes up with. And then one could say perhaps that there is a certain value in having, well, coming from different backgrounds and having the specific different interests that one has and convictions about what is valuable and what is not valuable. And then one could say that it's always important to acknowledge that there are different value statements, interests, and that it is not, and that would perhaps relate back to your point before, that in a way then aligning different activities too early or too strongly can cover up the way in which we also might disagree about certain value statements.
Thank you very much. I want to thank you both, and yes.
Okay.
I need to wrap up. Thank you very much for participating. Thank you for the audience, and see you tomorrow.
The last of the speakers is Jens Hauser. I guess many of you know him already. So he needs little introduction. I'm not sure here. Hauser. Yeah. Please.
Yeah, thank you for the kind introduction. That was very long. That's the thing when you are last and everybody is going to believe that you have been anticipating all what have been said in between. And I have not. I'm just here to play what Rodrigo asked me to play, devil's advocate, in something like 15 minutes. And of course, it's very interesting to see how much emphasis has been put on bottom-up biotechnology. I just want to flag up that there has been a lot of exhibitions that have been touching upon this issue, and for example here the synthetic exhibition about the ethics of synthetic biology at the Natural History Museum in 2011 that I organized with my colleague Markus Schmidt in Vienna. The Whiteware exhibition on art and synthetic biology we organized in last year in Los Angeles, precisely at the UCI. There was also exhibitions inspired and as a result of artist in residency there. We have been creating residencies, for example, at UCI for 2 artists working with non-canonical amino acids and macroscopic protocells that turn out to produce bioluminescent patterns as phytoplankton as seen from space. We have also created a residency in Denmark at the Flint lab in Odense that is run by Stine Rasmussen, one of the very basic chemists that we know as being involved in artificial life, but the chemical bottom-up side since the late 80s already. So we have already learned that sometimes art and science relationships are being put up because it's making science inviting and sexy and is attracting them attracting new talents. We also have known and learned that we shouldn't talk about innovation, and we shouldn't talk either about creativity. So of course there may still be researchers today in the natural science who are facing the artist operated with qualifiers such as beauty, who expect the artist to help them better visualize their findings. And there still may also be artists who, crossing the threshold of the laboratory, perceive a kind of army of technicians potentially inclined to produce and materialize their preconceived ideas, and we may all know such examples. I put fruitful misunderstandings and an epistemological turn in the title of my short intervention here today, because it seems to me that the misunderstandings do not only concern supposedly different expectations between what the anthropologist P.C. Snow once called the 2 cultures, but that the supposedly untouchable binary art science itself turns out to be a misunderstanding as such. So, First, the status of images. We know that images are efficient machines to sometimes generate fallacious metaphors. In these two opposite scenes of anatomy here, visual analogy is purely formal with regards to what is represented and with regards to the status of the image. In Rembrandt's anatomy, the image is the art, depicting persons in black habits looking at a human body being dissected, bathed in light that transforms the place symbolically into a place of knowledge produced by incisions in a dead carcass. On the left, it's an unpretentious amateur snapshot from the first biomedia art hands-on workshops for artists in London organized by Symbiotica and the Arts Catalyst in 2005. A group of artists in white lab coats learning gel electrophoresis techniques, cleaving DNA molecules with enzymes, and we see kinds of anatomy in both cases, yet everything changes. The status of the image, the role of the artist, the scale and status of what is considered a body. However, the assumption that an artist approaching a researcher in natural science would be first and almost interested in aesthetic images is still widespread. This autumn, at Michigan State University, where I also co-direct the artist in residency program Bridging Art and Science, if you want to. I consulted the biochemistry and molecular biology labs on campus for my personal research into greenness studies, quite logical, titled Ouvert, and we have another upcoming conference here, and there are some flyers around for the next Society for Literature, Science and the Arts conference that we organize precisely on the topic of green, ungreen and prismatic in Copenhagen in June, so please pick a flyer. That was a commercial break. So the very dear colleague David Kramer, who runs the center at MSU, thought to get straight to the point. Oh, you're interested in art. I have some very nice images here and beautiful time-lapse movies from my chloroplast research. And it took me some time to explain that despite the colorful and indeed spectacular images, the purpose of my visit was to borrow a number of portable devices, spectrometers that David had developed to conduct field studies at inaccessible locations, which allow real-time analysis of photosynthetic and protective metabolism in plants for art projects. Oh, you mean you really want to work with artists hand-on with my machines? So of course, the artistic research approach here was an epistemological one, because one of the crucial questions when measuring greenness with regards to ecology and climate-related modeling is not what we measure, but from where we measure. So example one, on the one hand, there are abstract remote sensing measurements of satellite-based normalized difference vegetation indexes, pixels of more or less uniform greenery to map carbon exchange data crucial to assess impacts of CO2 sequestration strategies. But here, vegetation is considered just monotonous greenery and quantifiable CO2 neutralizers within global carbon budgets, but while qualitatively neglecting biodiversity. On the other hand, we may obtain ground-based qualitative data accomplished with such handheld devices in a strategy of what feminist philosopher Donna Haraway has described as situated knowledge, a stance that inspires, as we know, many artists today. So more and more initiatives show this hands-on attitude of a shared experimentation of both concepts and technical devices, and it's gaining ground in this field of artistic research. When reading the interviews conducted with the first class participants, some patterns seem to be quite significant. Of course, here are some opinions from biologists. For example, what can I learn from artists? To be designers, they could help design our microfluidic channels. Why not? Public engagement, if the artist can help with our work, that would be useful. So kind of utilitarian approach to why an artist could come in. But also, I have learned to better explain my work to people outside my field. So explaining a world, so it's getting new skills through the artist. So all this is coming from the material that Rodrigo provided. Artists, for their part, think, for example, that I believe artists can certainly contribute for the advancement of science, a field that requires both imagination and creativity. And then we also have answers that go in a very different direction. A biologist seeing here a dimension that is maybe not often enough the focus of debate. One of the biggest temptations facing scientists today is the use of high-end technology instead of reason. If we are given a technical solve, we would rather just throw everything in a machine and see what comes out. A lot of artists have noticed this back and forth with technology. While a new technology can help us see something differently, it can also obscure or distract from its original intention. And this last point precisely is addressing the potential of an epistemological turn, not to represent scientific concepts and what we know, but to experiment hands-on how we know what we know, while sharing the tools and increasingly convergent technologies with a critical mind. This is also the opinion of philosopher of science Hans-Jörg Reinberger, who sees a much benefit from the humanities for the humanities as for the natural science to fruitfully address natural science media blindness and the uncritical metaphoricity because, quote, there's a general tendency on the part of scientists to blend out the epistemic dimension of the work, the ever-changing means and media. They tend to look through them, to view them as allowing immediate access to the findings. So in the light of convergent technologies, I find it useful to complicate a bit this alleged art-science binary. How can it be even today that we separate on the one hand the natural science and the engineering science and arts and the humanities at large, the Geisteswissenschaften on the other, which all the same also still include disciplines such as philosophy and sociology of science. At the same time, especially in the media arts, we find enormous technical understanding and competence. So how can one see the arts then still today as natural science, nearly natural other? So we find increasingly artists who, beyond knowledge production or illustration, or the visualization of scientific results, want to question these experimental systems that form hybrid epistemic objects, to come back to Hans-Jörg's terms again. And beyond theory and the researcher's own agency, these epistemic objects are made as well by the social structures of the lab, the material arrangements of model organisms, the instruments, in short, by the contemporary technology and media. And it is the latter that art and science share above all. Since then, there have been many artists in laboratory spaces, as there are laboratories in artistic spaces. And if you follow up the arguments of Bruno Latour when examining climate and weather engineering as humankind's collectively expanded meteorology between multiplied macroscopic processes and macroscopic effects, quote, the laboratory has extended its walls to the whole planet, so much that within this world of hybrid causality, in which natural and cultural forces can no longer be distinguished, even, quote, artists have perforce become white coats, who simulate, sculpt, stage even atmospheric phenomena. Agnes knows something about that. So the question is, what is precisely a laboratory today? Second, how beyond the usual symbolic and formalistic evocations, artists can generate system awareness for the invisibility of the microscopic, for the incomprehensibility of the macroscopic, for example, when working with biological in vitro systems. So, however, to counter this fashion that is this chic for an artist just to cross the threshold of a laboratory, which laboratorium, no? Laboratorio in Italian, for example, can just refer to a similar workshop or a craftsman or an artist. Oren Katz, the co-founder of Symbiotica, precisely evoked this artistic laboratory dedicated to the research, learning, critique, and hands-on engagement with the life sciences, has pointed to the vagueness of the term of the laboratory. An artist, even in a Leinsam's lab, can have extremely different attitudes, and one can do all and its contrary in the lab. So Katz differentiates, for example, the illustrator, the commentator, the visitor, the onlooker, the appropriator, the entertainer, the user, the industry worker, the hoaxer, we have a lot of them, the hobbyist amateur, the after the hours under the table, That may be where the biohacking comes in. The mail order or ready-made artist, very Duchamp's attitude, or the researcher embedded in a science technology setting. So I will now rush very briefly through some of the artist positions just to show how many different things you can do in a lab. So first example, very known, victimless leather. Material Stage is a techno-scientific device to practice cell and tissue culture, and artist Katz and Zur here effectively carry out hands-on co-culturing of human and animal cells over a polymer scaffold in the form of seamless miniature garments. The utopia to create leather without involving to kill an animal is, however, ironic. Do these clothes really do not make any victims? Since animal products such as fetal calf serum are required, the status of the victim is simply displaced. So in a very literal interpretation of Marshall McLuhan's sentence that the medium is a message, here the overlooked culture medium is in fact the message. The technological device confronted with the promising discourse functions as the technological trompe-l'oeil. Another example, what we created on invitation of the Schering Foundation and Heike Schering in Berlin in 2015. Sterile, Revital Cohen and Tur van Baalen have asked a Japanese biologist to produce goldfish for them that are engineered via a treatment with morpholinos to hatch without reproductive organs, born to be sterile, in an edition of 45 to very much cope with the white cube speech. In parallel, they constructed a machine that is reenacting the biologist's movements and actions so to potentially automate the production of sterile goldfish as a kind of design version of outsourcing of reproduction. So which kind of Oren Katz categories would this fall into? And who is taking responsibility if Mr. Yamaha, the Japanese biologist, actually commits harakiri? Adam Brown's aesthetic installation Origins of Life here presents what in the language of performance art we would call a reenactment of the famous experiments carried out in the 1950s by Miller and Array, intended to highlight the chemical origin of the appearance of life on Earth in a staged reduced atmosphere with a primordial soup. But during the artistic experiment itself, conducted for many years, the artist and his scientific collaborators, Robert Root Bernstein, found organic compounds, molecules not revealed in the original experiments. Here we have another case of pop artist Billy Apple, who has produced an immortalized cell line out of his own lymphocytes to become a resource for art and science, and at the same time granting himself as an artist cellular immortality. Very interesting example, of course, is Paul Van Nuys in this biotechnological installation we showed in Berlin, latent figure protocol. He destabilizes the idea we have from so-called genetic fingerprints, from the mindset of a tactical, politically motivated media art practitioner. He performatively deconstructs the very notion of what a DNA fingerprint might be, which precisely is not an imprint but a trace of the body which has been manipulated through laboratory procedures. It needn't come from the finger at all. By treating each line of the gel as a row of pixels composed of DNA fragments, figurative images emerge from a known DNA sample instead of abstract patterns from an unknown DNA sample, such as OD01, the copyright symbol, or the skull and crossbones. Hence materializing the eternal chicken-egg question in how far the scientific results are determined by the technical models of representation. So what Van Nuys is doing here is applied STS. He makes transparent sets of technical and biological non-human agency, thus emphasizing what scientific papers usually hide in the materials and methods section. So my argument here is that the arts and science dichotomy may be a shortcut in the light of overlays in the technical media. And avoiding these media and materials, which may be common ground, presents to risk of reactivating the residues of a binary division in research cultures between the humanities and the natural science, as illustrated as well in the late 90s by the so-called SoCal case, We remember that physicist Sokal and Brigmont accused postmodern philosophers of abuse of science and its technical terms, just producing vague fashionable nonsense, imposture intellectuelle, meant to unmask philosophers such as Virilio, Lacan, Kristeva, Latour, Baudrillard, or even Deleuze and Guattari in their alleged superficial erudition when using concepts beyond their competence, such as entropy, Gödel's incompleteness theorems, or quantum physics. But we also remember that in the following debates, bitter debates, the French philosophers and sociologists of science shot back, claimed the conceptual freedoms to appropriate, and blamed the authors of the pamphlet themselves for not paying attention to the fluidity and shifting metaphors that are often used literally and uncritically by natural scientists themselves. So my claim is that beyond visualization, sonification, data translation, such works have to be analyzed on the basis of the artistic media themselves, with their respective epistemic nexuses. So such cultural practices need to be approached by beyond an image-based hermeneutic approach. It is often here that fruitful misunderstandings emerge when mediation and technologies are not employed merely to achieve an aesthetic effect, but when they rather become themselves entire elements of the aesthetic idiom. And I'm coming back one last time to this biologist interviewed about the first-class residency, which I found very interesting. He said that in scientific research there can be an overemphasis. It's a kind of unfounded arrogance. I remember seeing a circular diagram in the Bauhaus in Dessau where science was a part of a larger wheel meant to inform design and society as a whole. This is somehow how I see science, integrated, informing and learning from other disciplines. And since the Bauhaus school is evoked, and since Natalie is here, I want to conclude with a quote, perhaps still relevant, from one of the Bauhaus protagonists, Laszlo Molinari. Not only was he a painter and a photographer, but also he was concerned all with how technology impacts on both the artist and the scientific's role. Quote, although the research work of the artist is rarely as systematic as that of the scientist, they both may deal with the whole of life in terms of relationships, not of details. In fact, the artist today does so more consistently than the scientist, because with each of his work he faces a problem of an interrelated whole, while only a few theoretical scientists are allowed this luxury of a total vision. The main difference between the problems of artists and those of scientists is the difference in the form and their materialization and grasp. In other cases of the past, scientific institutions have actually concluded that some extravagant assumptions may be more artistic than actual physics. So I'm grateful to be able to attend class first encounters and wish you all that you will well misunderstand each other. Thank you. Please see you all in Copenhagen in June.
Yeah. First of all, excuses for not introducing you properly, but yeah, I'm a little bit stressed. Does somebody has any questions? Yeah. Pablo?
Hi there, how you doing? Yeah, just a quick question. You pointed out a couple of points of view from artists or from scientists when they are visited by each other at the lab. And I was just wondering, have you come, or has anyone complained, or has anyone addressed the fact that the acquisition of data by scientists many times, I'm not going to say it's biased, but it's obviously elaborated towards, in order to fulfill, I'm not going to say to obtain the grant or to fall in the path of that scientific paper, or maybe your PI wants to go into that direction. many times data has probably been, it has to go through a matrix of acquisition that has already been pre-established. The equipment that we're building is, we're only measuring the fact that we want to measure in those cases. So has any of this, have you heard artists questioning the acquisition of the data, of the nature of the equipment or the nature of the analysis? Has that come in the discussions?
I guess yes. I have to really think deep in my memory if there are cases about data management as such. But for example, even if you look at Power News, it's very much about also racial profiling, the role of statistics, the role of how data collection, how the correlations are being built, on which kind of assumptions. So for example, in how far racial data is correlated with crime statistics and so on. That's why what we did in the installation in Berlin, the SIG, the Suspect Inversion Center, we actually created the image of O.J. Simpson's genetic fingerprint, but only using the artist's own DNA, so a kind of molecular photoshopping. So correlating also, going back to where's the data coming from, why have there been these DNA wars, in how far are the materials and the methodologies questions, or is it just the metaphor that takes over because it's a very handy metaphor to call it a genetic fingerprint, and the penny drops. Right? So I think there's more and more of this art, but I think we neglect a little bit this critical approach where I think both sides, if there are both sides, have something to win.
Hey, thank you for this very interesting presentation. I want to add something. I want to add another speak bubble onto your slides, because what I realized researching in the field of plant intelligence and plant behavior, that there is research that cannot happen, that is not funded because it's too radical for the scientific community, for the gatekeepers, and that's why it can only be done in an environment where there's a fertile ground. For instance, in the environment of the artist, they can do this research because the scientists cannot do this, because they're not allowed to, because they get punished when they do it. They don't get money. So, for instance, in Germany, you cannot get any funding if you want to do plant intelligence or plant behavior research. You have to go to Italy or maybe to Spain or to Australia. So in Germany, because, and the interesting thing is now, and the question I wanted to ask the guy who was talking about how science is, but he was not telling us who decides what is researched and what is not allowed to be researched. Yeah, so this is the big field where scientists can actually go to the art, into the field of the art, and then do their work there, which they cannot do somewhere else.
A good example of that, because you're mentioning plant research, we just concluded a kind of very inspirational gathering in Paris at the École Polytechnique, together with the École Nationale Supérieure des Arts et Design. So we've got €1 million recently from a foundation to conduct art and science relationship for the next 5 years. And the first event of the series was called Devenir Plante, becoming plant. So it's kind of extension of the Deleuzian and Guattarian principle of becoming animal, becoming mineral, becoming whatever. And but this plant alterity thing is very interesting. And there was some researchers also, I think, who explained their work by not precisely calling it plant behavior studies or plant neurophysiology, but who have to invent other terms. So it's in fact not the research itself, I think, that is at stake, but how you frame it, how the metaphoricity is valid to funding bodies and whatnot. So there were a lot of researchers, I think, that have benefited also from the artist's presence in the lab. There was artists doing workshops at École Polytechnique, which is completely crazy because it's a kind of military complex. So it was a kind of total disruption of the center, you know, but also providing them with new metaphors, right? So, for example, you can talk about plant intelligence, but you can also see you will focus on plant volatile signaling, you know, and then you have the same thing. It's not that you have to do other research, I think, but you have to phrase it differently. So it's again the question of metaphors and the power of metaphors. And there also I see a large potential for artists actually, maybe by even sometimes overdoing, stretching to the extreme can also be helpful.
James, because really so now I'm very happy to welcome Beatriz Simoes and Rayan, a colleague from the Institute, that are going to interpret a few pieces to, I don't know if to relax or to just completely change these very dense talks that we had had. After that there will be some wine outside and don't know if I'm a little bit.